and then he moved to the U.S. in New York, and he has been staying in Manhattan for quite a long time. So he did his PhD in Columbia University under the guidance of Dr. Richard Mann, a fly person that works on developmental biology, and especially in Hox gene, and that's what he worked on during his PhD. And then moved on to Rockefeller University in Dr. Herman Steller's lab, who is famous for his work in apoptosis. In 2005, he became an assistant professor in cell biology in NYU, which he started actually working kind of shifting gears from apoptosis to other stress signaling pathways, including ER stress. And also he's going to be talking about this integrated stress response, which is related to a lot of neurodegenerative diseases in aging. He has risen up the ranks to assistant to associate to full professor in 2020. And he's been really involved in both education and research mission of the school as well. He actually is one of the co-directors of the graduate program. A number of his trainees have gone on to have their own laboratories. And it's really a great pleasure. So I've only met him once during a fly meeting, just say, hey, hi. But I wanted to come to say hi because we have been getting a lot of reagents from him. And I think what Dr. Ru is really known for his generosity, generating really very useful reagents, antibodies, reporter construct mutants, and really sharing that with the rest of the community. So it's really, it's a tradition in the fly field, but he really has that spirit. So please help me welcome Dr. Ru to the podium.
Okay, let me just, can everyone hear me? Yes. Okay. So thank you for that kind introduction. And in terms of sharing, generating useful materials and sharing it with the community, I mean, I should thank the investigators here at the Baylor College of Medicine, especially Hugo Bellin, for generating tremendous amounts of useful materials, without which I won't be really giving this seminar presentation. So, and thank you for inviting me. Okay, so let me get started. So today I'll tell you about our work on the stress responsive signaling pathway called the integrated stress response. And I chose this title, Found in Translation, because it's going to have a focus on mRNA translational mechanisms. So let me start by telling you why we think mRNA translation is important in this stress responsive signaling pathway. So this is a cartoon view of a ribosome translating an mRNA. So this is supposed to be the mRNA. This is supposed to be the nascent peptide. And if you asked me 30 years ago, I mean, I'm interested in like phenotypes in the context of an organism. And if you asked me 30 years ago, I would have said, oh, mRNA translation is boring to me, because it's just something that occurs constitutively in cells. It's never going to give rise to like specific phenotypes. And that view has really changed, I think, in the past 30 years. So let me just tell you a few examples. All right. So when we have lots of nutrients, or for example, nutrients can affect the amount of growth hormones released in our body, and then we become big and strong. And an important contributor to that process is nutrients and growth hormones somehow affecting signaling pathways. And I just listed two, but there are many. Insulin signaling pathway, TOR signaling pathways. And people who investigate these signaling pathways report that an important outcome of these pathways are stimulation of mRNA translation. Okay, and then cells become big, and those animals or experimental animals, the body becomes big. But then there's this observation that many people are reporting. Growth, generally, there's an inverse relationship between growth and cellular quality control. And just to show you like a photo of a celebrity to make that point, is this is a photo of a man named Andre the Giant. He was a celebrity in the 1970s and 80s. And he suffered from this disease called giganticism, which basically means that this individual's pituitary gland is expressing too much growth hormone, and growth hormones eventually affect our livers to release IGF-1, which gets released to the rest of the body and activates insulin-TOR signaling, and they stimulate protein synthesis and growth, and therefore these people become big and powerful, but then they are generally thought to be weak in wear and tear, and people suffering from this disease generally suffer from age-related diseases such as diabetes, heart disease, and many others. And this particular individual also passed away in his early 50s due to heart attack. I mean, this is an anecdote which scientists don't really like, but just to illustrate the fact I'm conveying the story. And also many people are reporting that the converse is also observed, especially if we have less nutrients or if we secrete less growth hormones, then they're going to have the exact opposite effect, and also they'll activate stress response pathways, and many stress response pathways, including some that I'll mention today, they act on the mRNA translation machinery, generally reduces protein synthesis. And an example of this is are these people referred to as Laron dwarfs. So these individuals have mutations in the growth hormone receptor. So their bodies think that there's too little growth hormones coming from the pituitary gland. And because now growth hormones affect IGF-1 release, and therefore these people, their cells are going to think that there's generally very little insulin signaling and TOR signaling, and their cells grow less, and their bodies are small. They actually suffer from lots of developmental diseases, but this Ecuadorian doctor that has been following these patients, the same patients for the span of 20 years, and the results are published in this paper published 10 years ago, report that one thing these people benefit from less growth hormone, insulin, TOR signaling is that they have very scarce incidence of age-related diseases, including diabetes, heart disease, cancer, neurodegenerative disorders. So these are just some of the really large body of work out there indicating that there is a generally inverse correlation between growth and cellular quality control. But then when I say this to a scientific audience, of course, many people raise eyebrows because what I just said is just correlation. It's not a controlled experiment. How can you be so assertive? And I fully acknowledge I used anecdotes and correlation here, but the rest of today's presentation is going to be about controlled experiments, focusing on specific genes to establish causation. And I will focus first on a protein synthesis inhibitor called 4EBP. Okay, so let me explain what this protein does. Before that, I'll just give you like a very simple view of mRNA's translation. So shown here is an mRNA, right? And they're different from tRNAs and rRNAs in that they have to eventually load ribosomes. So what does the mRNA have that tRNAs and rRNAs don't have? And that's one of the features is they have a unique structure at the 5' end. It's called M7 guanosyl residue, and a very unique conformation, and I'll refer to that as the 5' cap. And this 5' cap is really important for mRNAs because that is recognized by a translation initiation factor called eIF4E, which, and some people don't like this literature because there are lots and lots of factors involved, but to really simplify things, they eventually, through the help of many other factors, recruit the small subunit of the ribosome, which comes with initiator methynyl tRNAs, so that they can start scanning for AUG start codons. And once they find an AUG start codon, they're going to recruit the large subunit and start protein synthesis using initiator methionine tRNA as a starting point. And 4EBP is a protein that binds to EIF4E, that's where the name comes from, and inhibits this process. So again, if you asked me 30 years ago, in the future, do you want to work on this gene 4EBP? I would have said no, because back in 30 years ago, the general view was that virtually all cellular mRNAs undergo this EIF4E dependent translation. So this is going to be a constitutive indiscriminate process. So I won't be seeing any specific phenotypes, but that view has changed a lot in the past 25 years or so. Okay. Now, something's not working. Yeah. Okay. All right.
So, yeah.
Okay. Enter. And that view has started changing around 25 years ago when people started finding that nutrients activate, for example, signaling pathways that activate this protein called mTOR, and in flies, we call it just TOR. And an important outcome is so when TOR is active, it stimulates cell growth. And how does it do that? It stimulates overall protein synthesis. And how does that do that? It phosphorylates and inhibits the inhibitor of cap-dependent translation, 4EBP. So this thing is apparently being regulated. And that's not the only story. All these different laboratories, maybe a little less than 20 years ago, have been identifying 4EBP transcription as a target of a signaling pathway mediated by FOXO. This is a transcription factor. And FOXO responds to lots of different types of stress. And this is partly why if you give cellular stress of various types, then cells reduce their protein synthesis and reduce growth. And at the same time, now they jack up their resistance to various stress responses. So this initially, when I started encountering this literature, I found it a little bit odd because it doesn't fit with our current understanding of what EIF4E did and what 4EBP did at the molecular level. I mean, these are supposed to be having indiscriminate effects. Let me just introduce one other study. I mean, there are actually many studies along these lines, but using model organisms like Drosophila, people have been noticing that when there is some stress, for example, FOXO activation or reduced protein intake, let's say reduced yeast levels in the food, and if it's mild, if it's really strong stress, then the flies will die. But if there's a mild stress, then there are certain mild conditions that would make flies live longer. And for example, this paper is arguing that that kind of lifespan extension is happening because these conditions induce 4EBP, and 4EBP helps with stress resistance and helps with preventing wear and tear. And aside from this paper, there have been large numbers of papers reporting that if you just force express 4EBP, then flies do a lot better in response to various different types of stress. Okay, so, and when we got interested in this particular problem, at least with one type of stress, reduced protein in the diet, what is the signaling pathway that activates 4EBP transcription was not known. And at the same time, we were working on a pathway that might have something to do with reduced protein in the diet, and that is the pathway referred to as the integrated stress response. So why is it called the integrated stress response? That's because there are many different types of stress that converge into a common signaling outcome. For example, this is the pathway described in mammalian cells, and mammals really don't like long double-stranded RNAs. And that's because when they see long double-stranded RNAs, then they activate a stress-activated kinase called PKR, and it really, the cells start going all right because this pathway is abnormally hyperactivated. When there's problems in the endoplasmic reticulum stress, then it activates a different stress-activated kinase called PERK. When there's amino acid deprivation, then again, it activates yet another different kinase called GCN2. And if there's mitochondrial stress, then in the past several years, groups have been reporting that there are some mitochondrial proteins that get released to the cytoplasm and activates this kinase called HRI. And once these kinases get activated, they phosphorylate the same target. And that target is a translation initiation factor called EIF2, the alpha subunit. Okay. So once this happens, because now EIF2 is an important translation initiation factor, protein synthesis is reduced, then already it's thought that that would help some of these stressed cells. Because, for example, when there's not enough amino acids, you don't want to use up all the limited sources of amino acids. So it makes sense that cells reduce their protein synthesis. When there's not enough chaperones to fold proteins in the ER, you don't want to put new nascent proteins into that environment. That would make things worse. So already, this is thought to provide some kind of a homeostatic protection. But then there's something very weird happens, which is most protein synthesis is reduced under these conditions of stress, but then there's a transcription factor whose synthesis actually paradoxically increases under those conditions. And I'll describe that mechanism a little later. And this transcription factor now induces various stress-responsive transcripts to help out the cells. Okay. And in flies, at least 2 of these kinases are conserved. So we'll be dealing with this pathway for the rest of today's presentation. Okay. So what kind of genes do ATF4 induce once there's the stress response pathway is active? And already, I mean, there's lots of literature out there, especially in mammalian cells, reporting what kind of genes are induced by the integrated stress response. This is just our version. We also did gene expression profiling in the fly, and I'll get into the technical details later, but there is a tissue with physiological integrated stress response, and that is called the larval fat body, and I'll show you the evidence later. But suffice to say, for now, we did ATF4 dependent gene expression analysis by knocking down ATF4 in this fat body, and this is a volcano plot. And the first class of genes that really drew our attention is, cytochrome P450 genes are really dependent upon ATF4 in this tissue. And to us, that indicates, well, ATF4 in response to stress, now it wants to enhance stress resistance in these cells. Of course, they want to induce cytochrome P450 and other antioxidant genes that would help get rid of oxidized molecules or foreign molecules, damaged molecules, flush them out of the system. So maybe this is why this pathway helps enhance stress resistance in these cells. The other class of genes that really stand out are genes involved in amino acid biosynthesis. So shown here are, for example, so there are essential amino acids and non-essential amino acids. And this pathway is activated by amino acid deprivation. And by this, seeing that there are many amino acid biosynthetic enzymes that are under the control of ATF4, we can already guess, yes, this must be a homeostatic signaling mechanism to enhance non-essential amino acid biosynthesis when there's not enough amino acids in the diet. So here are some of the other examples. Enzymes involved in serine biosynthesis, glycine biosynthesis, glutamine synthesis, synthase, and serine and glycine, some of these enzymes particularly draw our attention in terms of stress resistance because one of the important antioxidant system in all of our cells are glutathione-mediated detoxification of oxidized molecules. And glutathione requires enzymes like glycine, et cetera, et cetera. Okay, so we think that the integrated stress response has really evolved in large part because this is an important homeostatic signaling pathway that helps express genes that would help reduce the burden in the endoplasmic reticulum and cope with amino acid deprivation. Now, in the first part, as promised, I'll tell you something about 4-EBP. And 4-EBP, I'll tell you that 4-EBP is a transcriptional target of ATF4, and I'll discuss what the implication of having a cap-dependent translation inhibitor is within this pathway. And in the second part, I'll talk about this paradoxical induction of ATF4. Again, there's a translation inhibition step, but then ATF4 is never synthesized when other proteins are synthesized really well. It only gets synthesized when all other protein synthesis is compromised. So what's going on over there? We'll discuss the mechanism. And then in the last part, I'll tell you that this pathway is, of course, just like anything else, things are more complex. And it appears that there are other branches of the integrated stress response. And one of them we have recently identified. So we'll talk about that. Okay. So part one, we'll talk about 4-EBP. Decline? Approve? Decline? Oh, okay, all right, okay. All right, okay. Okay, so enter. Okay, all right, okay. I got to the next slide. Okay, so we're gonna talk about 4-EBP. Again, 4-EBP is a protein that binds to EIF4E, and therefore people like to refer to this as inhibitor of cap-dependent translation. And I just told you earlier that 4-EBP can be induced by stress. I talked about starvation, but it also responds to other types of stress, for example, bacterial infection. And all these studies which preceded ours reported evidence that 4-EBP can enhance stress resistance in model organisms. Okay, all right, okay. Maybe I need to press it a little longer. Okay, so the start point of this was, actually the reason why we got into it was a little more convoluted, but Minji was a postdoc in my lab already a while ago, and she got us interested in 4-EBP. Really, her initial motivation was she wanted to work on FOXO because it looked like an interesting gene. But then all the FOXO experiments gave her only negative results. And then we decided to step back, okay, all these literature saying if you feed flies less protein and 4-EBP is induced, well, maybe it's okay it's not FOXO, because the integrated stress response that we study, I mean, it also is supposed to respond to amino acid deprivation. So she was disappointed about the negative results with FOXO, but she decided to test this alternative model, and it gave her a positive result. So this is qPCR result from adult flies. If you starve them and do qPCR from the intestine, then 4-EBP is induced. Okay, lots of other people have reported this before. But then when she knocks down GCN2 or ATF4, then such induction is really impaired. So it seemed like she was on the right track. Okay, And most recently, I mean, we've been doing, why not just do RNA-seq? And in fact, we do RNA-seq and we can validate Minji's old result. When we knock down ATF4, 4EBP is also reduced. These results are substantiated. And Minji went on to demonstrate that this is 4EBP is a direct transcriptional target of ATF4 because she can see predicted ATF4 binding sites within the intron of 4EBP. And when she takes out this DNA fragment and hooks it upstream of DS-RED, then the reporter now starts responding to amino acid deprivation. These are cultured cells and without grown in methionine deficient media, and the reporter is induced, but then this is ATF4 dependent because when she knocks down ATF4, then the reporter fails to be induced by methionine deprivation. And when we make flies, then we see this reporter activity pattern in this red, which actually appears more like orange because of the green background, like this. And okay, many of you may be unfamiliar with Drosophila larvae, but maybe at least the people who grew up in this country may have read the book Very Hungry Caterpillar. And there you'll see that the larvae, when they're little, they're in the food, eating all the time. So these small larvae, they have plenty of nutrients, and the reporter activity is suppressed. But then late third instar, this is when the larvae are feeling, well, they're full, and they crawl out of the food, and then they eventually become pupae, and perhaps because they're now starting to experience amino acid deprivation, this reporter starts lighting up in this particular tissue called the fat body. And we know that this is dependent upon ATF4 because when we take these third instar larvae and we knock down ATF4, then the red reporter disappears. So this red reporter is a readout for ATF4 signaling, physiological ATF4 activity during development. And at least in this old study that got us interested in 4EBP, Minji decided to follow up on the previous literature on 4EBP. So all these previous studies hinted that 4EBP affects lifespan. And some have argued that dependent upon the nutrient status. So she did lifespan experiments in either control or GCN2 mutants. And what she sees is she found a condition based on the literature that if she reduces the yeast content to a certain degree, then the flies actually live a little longer, statistically significantly, shown in red. And such beneficial effect of reducing yeast in the diet disappears in the GCN2 mutant background. And this is pretty much similar to the reported 4EBP mutant phenotype. So we felt that we were on the right track. Okay. But now there's a challenge. I told you on paper, none of this makes sense because EIF4E dependent mRNA translation, people were saying virtually all mRNAs undergo EIF4E dependent translation. So 4EBP should inhibit every mRNA translation, but then we're getting this specific phenotype. So how can that be? And I mean, if anything, 4EBP, ATF4 is trying to help the cell and it induces lots of transcripts. But if 4EBP inhibits the translation of these stress responsive transcripts, then it would be detrimental to stress rather than helping it. So something wasn't really making sense. And around this time, Deepika Vasudevan was another postdoc who came to my lab, and she, as her first project, she decided to tackle this particular question. So her entry point is now using a slightly different model. So there was a study, one of the first studies on Drosophila 4EBP. This paper reported 4EBP. 4EBP's Drosophila name is Thor. And this group reported that without 4EBP, flies become vulnerable to pathogenic bacteria. Okay, so it's an innate immune response gene. And then years later, a totally different group published a totally different paper, probably not even aware of this earlier study, reporting that GCN2 contributes to innate immunity. And when we saw these two papers, Deepika felt she knew how to fill the gap, and she demonstrated that it's actually GCN2, pathogenic bacteria somehow activate GCN2, which activates ATF4, which induces 4EBP to help with innate immune response. But again, how does 4EBP help with innate immune response? It doesn't make any sense, because innate immune response requires the synthesis of antimicrobial peptides. So this is just one of them. There are many of them, but we're using this drosomycin as a model. So drosomycin transcripts are induced, and they have to be translated. And at the same time now, these other groups are reporting that this translational inhibitor 4EBP is induced. If this is inhibiting the translation of drosomycin and antimicrobial peptides, then 4EBP should be a factor that interferes with innate immunity, but these papers are arguing that they actually help with innate immune response. And based on this, Deepika decided to test the hypothesis that maybe these innate immune response genes can evade suppression by 4EBP. And because now we're talking about, cap-dependent translation at the 5' end, maybe there's something special about the drosomycin 5' UTR. And she decided to do, among other things, reporter analyses and drosomycin promoter, driving drosomycin protein fused to GFP, but either with its own natural 5' UTR or a generic 5' UTR from tubulin. And she's seeing that pathogenic bacteria induces this antimicrobial peptide only when it has its own 5' UTR, but not a generic 5' UTR. There's something special about this 5' UTR, and this is quantification, basically, the relative amount of drosomycin GFP protein compared to its mRNA. And you can clearly see that when it has a generic 5' UTR, then it's being suppressed by 4EBP. The translation is being suppressed. So what's so special about the antimicrobial peptide 5' UTR? And an obvious candidate back then was the possible presence of the internal ribosome entry site. So in that world, mRNA translation world, now things have become a little more complex, but back then, people were saying either it's EIF4E dependent, cap-dependent translation, and if that's not it, then there are these internal ribosome entry sites often found in viral mRNAs. And how do they measure internal ribosome entry site activity? Well, they do these things called the bicistronic assay, where there are two reporter proteins, and intervening the two reporter protein is the 5' UTR of our interest, and the logic goes something like this. So the ribosomes would load to the 5' cap, right? This is what happens in almost all cases. And then it translates GFP, and once it hits the stop codon, the ribosomes are supposed to fall off. The termination machinery would just tear them apart. So normally DS-RET never gets synthesized unless there's a special sequence over here that now recruits ribosomes independent of the 5' cap. And that's called the internal ribosome entry site. And when there's the internal ribosome entry site, then DS-RET can be translated. And in fact, when Deepika generated these constructs, what she found was that when drosomycin 5' UTR is inserted here in the correct orientation, then she can see the second cistron being translated, supporting this idea that innate immune response transcripts such as drosomycin has internal ribosome entry site activity. So, what do we think is going on? So I told you that all these different types of stress, reduced protein in the diet, pathogenic bacteria, they activate this pathway, and ATF4 tries to help the cells by inducing lots of stress responsive transcripts. But now among them is 4-EBP, and how does a translational inhibitor like 4-EBP help with stress response? And it's going to suppress many cellular mRNA translation, but stress responsive transcripts such as streptomycin has alternative ways to recruit the ribosome, so they'll at least not be inhibited by 4-EBP. And perhaps because now a lot of cellular mRNAs are not undergoing active translation, they might even have more resources available to more efficiently translate these stress response transcripts. So that's the working model that can be derived from our studies. Okay, so let's move on to the second part. Again, this ATF4 transcription factor involves translational inhibition, but then the stress responsive transcripts use alternative ways to get synthesized. So more or less it's a similar theme, but mechanistically it's very different. So now I'm depicting this pathway in a slightly different way. This can be GCN2 PERK. It's a stress activated kinase. It phosphorylates EIF2. And I told you, I don't know if I told you, but EIF2's normal role is to bring initiator methionine tRNAs to the ribosome. And that activity gets inhibited by the stress activated kinase. So most other cellular mRNAs, I mean, everyone needs initiator methionine tRNAs to start translation. So translation is going to be suppressed. But this ATF4 mRNA is weird in that when translation is efficient for all the other mRNAs, ATF4 never gets translated. And when EIF2 is phosphorylated and everyone else's translation becomes suppressed, ATF4 gets translated. And the key is in the 5' leader sequence. I'm going to say leader. I mean, it's the equivalent to 5' UTR, but maybe that term is a little bit inappropriate because there are small open reading frames that get translated, okay? So how does that work? Let's say the ribosome comes to the 5' cap with the help of, and it has the initiator methionine tRNA and it starts scanning. And now the problem is the first AUG that it encounters is not that of ATF4, but this decoy small open reading frame. So it ends up consuming its initiator methionine tRNA. But in order for ATF4 to be translated, this ribosome has to continue to scan because this 5' UTR doesn't have internal ribosome entry sites. So it has to scan and it has to eventually reacquire initiator methionine tRNA. And if translation is really efficient and that recharging is really efficient, then it acquires the initiator methionine tRNA early, and then it encounters yet another decoy upstream open reading frame. So because it's going to translate the decoy upstream open reading frame, ATF4, which overlaps but is in a different reading frame, doesn't get translated. So that's why when everyone else is happy, ATF4 is very unhappy, okay? But then when EIF2 is phosphorylated and something is becoming inefficient, the recharging kinetics is being delayed, and if the scanning ribosome gets loaded with initiator methionine tRNA after bypassing the decoy upstream open reading frame AUG, then now ATF4 has an opportunity to be seen, right? That's the model really developed by Alan Hinnebusch and David Ron and Ronald Dweck and several others, and is largely well accepted in this field. Okay. So this is what we're dealing with. And Deepika and I thought, well, that's a really complex mechanism. And, maybe EIF2, we can't explain everything through EIF2. There has to be additional regulators. So she decided to do a screen. And our advantage is that we have this really sensitive ATF4 reporter, right? And this is the pathway now when there's EIF2 alpha kinases and there's low availability of initiator methionine tRNAs, then things happen and ATF4 gets synthesized, and we can visualize that with our 4EB PDS strip reporter. So she started doing the screen. Now this is a control. Now she has her red reporter that reports ATF4, and a control reporter that doesn't care about ATF4. It's just constitutively expressing the green fluorescent protein. And so they normally, if nothing's happening, the larvae appear yellow. But then when she knocks down ATF4, then she specifically abolishes the red reporter, so they look like this. And in her first round of screen, she identified as a hit, a gene, an RNAi line that targets a gene that we had never heard of before, and that had the name of EIF2D. Okay, so this is just, after seeing this, we dissect the fat body, and you can see that the red reporter that responds to ATF4 really reduces its signal in ATF4 knockdown and also after EIF2D knockdown. So I told you that the business end of the ATF4 regulation occurs in the 5' leader sequence. So we generated a reporter. It has the ATF4 5' leader, or UTR if you will, but instead of driving ATF4, now it's driving a nuclear localization sequence tag DS-RED. And Deepika repeated the experiment because this reporter has a nuclear localization sequence. Now, as you can see in the fat body, nice nuclear localized reporter, but that reporter really disappears in the EIF2D mutant background. And Deepika found that EIF2D is not the only game in town, right? So again, what is EIF2D? I guess I didn't explain that. So it's a poorly characterized protein. Before we got into this, analyzing this gene, it was largely characterized by biochemists who did in vitro experiments. And they gave this name EIF2D because in a test tube, it behaves like EIF2 in that it can bring initiator methionine tRNAs to the ribosome in the test tube. But at the sequence level, it's totally different. And I guess the convention in this field is that if the biochemical activity is similar, then they give the name EIF2, and then there's A, B, C, D, etc., etc. So the sequence looks very different from EIF2, but EIF2 has domains that bind to, for example, tRNAs. This is the EIF2 protein. And a tRNA tail is making this contact with a protein. And there are cousins of EIF2. So EIF2, there are proteins with a PUA domain called MCTS1 and SUI domain called DNR. And the reason why I'm mentioning this is because Deepika found that there's some redundancy genetically. Okay, all right. And that can be seen over here. Again, this is the ATF4 reporter in a control background. Now, she adjusted the gain so that it looks like in EIF2D loss background, there's a mild reduction of the reporter activity. Then R mutant, there's only a mild reduction, but if she gets rid of both, then virtually all signal in the fat body disappears, indicating that these two genes play redundant roles. And now we're doing fly experiments. So reporters and looking at Western blots. I mean, many people can argue, oh, I don't believe that stuff. Flies, we can look at phenotypes. Are the gross morphological phenotypes similar? So we decided to look at that. And ATF4 was originally named by others before they knew that it was an ATF4 homolog as Cryptocephon in the 1940s. Can you read German? It says lethal factor. So it's a lethal mutation. Back then, you can publish a paper just showing this. And the phenotype is basically the head develops in the thorax, and then during the pupal stage, they evert to make the adult structures. And this eversion doesn't happen in the Cryptocephon mutants. Okay. And we repeated those experiments. So this is a wild type fly, pupal case, we dissected them out, and the flies have two eyes and the head and legs extending over here, wings. And this is the ATF4 mutant. The head is still somewhere buried in the thorax, so you don't see the head. And then on top of that, now the legs don't really extend. And what Deepika, and by this time now we had a collaborator who looks at the pupil phenotype all the time, Arash Vashula at University of Wisconsin. And he did this particular, his group did this particular experiment showing that in DEN-R mutants, okay, there seems to be a little bit of a problem in the head and the leg, but in the DEN-R and EIF2D double mutants, now they start looking very similar to the ATF4 loss, indicating that this is really probably a major regulator of ATF4 signal, these two genes. And these genes, their activities are conserved across phyla. And we know that because Deepika repeated this experiment in a human cell line called HAP1. And she first tried ATF4 induction experiments in the EIF2D knockout cell line. So basically, happy cells, they have ATF4 mRNA, but they have almost no protein. But when she challenges them with ER stress imposing drug tunicamycin, now this pathway gets activated and she sees lots of ATF4 protein induction. And in the EIF2D mutants, this appears to be compromised. And when she, in this EIF2D mutant background, if she further knocks down DEN-R, then ATF4 protein fails to be induced in response to the stress-causing drug. So things are conserved across phyla. And what do we think is happening? What exactly is EIF2D doing? And we're making an argument based on genetic analysis, and probably the mRNA translation experts are going to demand that we become test tube biochemists to really prove the point. So I'm just going to say it's not proven at that level. But what we think is going on is, as I told you, ATF4 induction requires this small scanning ribosome to acquire initiator methyl tRNA in a delayed manner. That's the key. Now, everyone has been thinking in the field that it's delayed because EIF2D is moderately suppressed by phosphorylation. So there's a moderate reduction of its activity. But maybe something that is worth considering is maybe the thing that ultimately delivers initiator methyl tRNA to this scanning ribosome in a delayed manner is not EIF2 itself, but its cousin EIF2D, and that's why this gene is essential for the integrated stress response. Okay, let me move on to the final part, additional branches of the integrated stress response. Okay, so again, this is the pathway that we're talking about. the integrated stress response. And now I'm going to switch the model itself. So far it had a lot to do with amino acid deprivation and GCM2 activation in the fat body. Now what I'm going to do is use approaches that impose stress in the ER and activate PERK. And so how can we, what model are we using? And we've been using these rhodopsin mutants that serve as a model for a human disease called retinitis pigmentosa. Right? So many mutations in proteins that impair their folding properties, they underlie lots of degenerative diseases. I mean, you can Alzheimer's, Parkinson's, the list goes on and on and on. Also, rhodopsin mutations cause age-related retinal degeneration. And lots of mutations have been identified, and they're not null mutations. They're usually, really the biggest category are the mutations that impair their protein folding property. Now, why does that have anything to do with the endoplasmic reticulum? Well, endoplasmic reticulum, shown here, is studded with ribosomes here, right? And the reason why they're studded with ribosomes is because virtually all membrane proteins and secretory proteins are synthesized on the ribosome studding lining the ER. And then once the nascent peptide enters the ER, they have to be folded. The first thing that has to happen is to be folded by the chaperones. And if you have a condition where a highly expressed membrane protein has a deleterious missense mutation, then it's going to overwhelm the cellular protein folding capacity in the endoplasmic reticulum. So it'll cause stress there. But actually, our animal cells are pretty good at dealing with these unfolded proteins. If you think about all these age-related diseases caused by structurally defective proteins, I mean, age-related means that when you're young, you're okay, even though those proteins are expressed at high levels. Why are young people okay? There's probably a robust quality control mechanism against that. Now our model is to take this one particular tissue, the eye imaginal disc, and we can express this mutant rhodopsin in this domain. And we're seeing that this mutant rhodopsin is now inducing ATF4, which is the integrated stress response transcription factor. So this is the model that we're using. And the story that I'm going to tell you is a work done by Brian Brown, who was an MD PhD student until recently. And he wanted to do gene expression profiling experiments. So the particular approach that he took was he just wanted to do gene expression profiling in the cells, in vivo tissue cells that he was manipulating. So he had to isolate those cells. And how do you isolate those cells? He decided to co-express with whatever he was expressing there with a nuclear envelope tagging GFP protein. And once now he's expressing whatever he wants to with this marker, and he meshes them, then now he can isolate the nuclei of those cells, and then now it becomes cell type specific gene expression profiling. And shown here is an image. Here's one nucleus with the GFP, nuclear membrane GFP, surrounded by all these beads, anti-GFP beads. And he did the sequencing, and lots of antioxidant genes named GSTDs as top inducers by this mutant rhodopsin. Okay, so what's up with this GSTD genes? And again, he formulated a hypothesis that turned out to be wrong. But the hypothesis is, well, GSTD genes, they're studies have pinpointed this region as the regulatory sequence. that make cells induce these GSTD genes in response to reactive oxygen species. And those studies had really named this NRF2 homolog CNCC as the gene that induces all these genes, again, in response to reactive oxygen species. So Brian thought his thesis title was going to be like, no one knew this, but CNCC is a mediator of the unfolded protein response. And he did a ton of experiments, and every single experiment was a negative result. So he was a little upset. But first thing that he did was, okay, let's first validate the results. So he took this GSTD GFP reporter, the enhancer fused to GFP, and he could see that, yes, his RNA sequence rhodopsin expression pattern is shown in red, and GSTD GFP is induced, and you can see that in green. So, all right. Because all the results were negative, let's again take a step back. ER stress responsive gene. So he tested it in the PERK mutant background. And what he found was that in the PERK mutants, mutant rhodopsin, shown in red, fails to induce GSTD, GFP in green. Okay, so it seems like PERK is mediating this response. And by this time now, he's saying, well, what's PERK's effector? I mean, you have to have a transcription. The obvious transcription factor, ATF4, again, is giving him negative results. So this is desperate, and then we just decided to test whatever is in the literature. And at the very last moment, one guess based on the literature ended up working, and that is a transcription factor that is associated with this disease model caused by ribosome mutations. So there are several groups studying ribosomopathies, right? This ribosome subunit mutations cause various diseases in humans, and implies that one copy ribosome subunit mutation also causes a certain phenotype. And people were reporting that this enigmatic transcription factor called XBP1 is mediating that response in their disease model. And I guess we were very lucky. I mean, first, XRP1 is a BZIP. And I guess we were very lucky because XRP1 mutants abolished ER stress mediated GSTD induction. So shown here is the experiment. High imaginal discs not expressing anything. Those expressing mutant rhodopsin, they're now inducing GSTD GFP. And when we get rid of XRP1 in mosaic clones, then within the mosaic clones now GSTD GFP fails to be induced by mutant rhodopsin. Okay, so all right, so there's this pathway, but I told you not any transcription factor can be PERK effectors because it suppresses mRNA translation, and ATF4 can be a PERK effector because it has a very special 5' leader sequence. And what she's finding is that, yes, XRP1 also has a similar 5' leader structure with these upstream open reading frames. And when she mutates, so when she's expressing these reporters now driving DS-RED, if it's a wild type reporter, then although there's a ton of mRNA, the protein never gets synthesized. But when she mutates a second upstream open reading frame, then the reporter gets lights up. And if she takes the wild type reporter, which doesn't light up normally, and overexpress mutant rhodopsin in green, then the red reporter starts lighting up, indicating that this 5' UTR reader sequence is really playing a regulatory role. So let's wrap up here. What have I told you? Okay, there's a generally inverse relationship between protein synthesis and growth. Inhibitors and stress resistance, and really I started and growth promoting pathways really inhibit this 4-EBP to enhance protein synthesis. And on the other hand, stress responsive pathways, they enhance 4-EBP transcription, and that probably suppresses growth, but then enhances stress resistance. And that's probably because many stress responsive transcription factors, transcripts have alternative ways to bypass these translational blocks. And that's why they fare better. And I told you about our work on the ATF4 induction mechanism, and also another branch of the integrated stress response mediated by XRP1. So with that, I'll conclude. These are, I tried to acknowledge people along the way. Brian Brown is here, Shahana Mitra is here. Deepika is not in the photo because she's now an assistant professor at the University of Pittsburgh. And Minji, she became independent a while ago and is professor at Ulsan University College of Medicine in Korea.
We're running a couple of minutes late.